An infection is the invasion of microorganisms in the body that are not normally present. The 4 different types of microorganisms that create infection are bacteria, viruses, fungi, and parasites. Bacteria are single-celled organisms that are responsible for illnesses like pneumonia and urinary tract infections. Viruses are not living organisms as they rely on a host to reproduce, causing illnesses such as a common cold, measles, or coronavirus. And fungi can cause skin diseases such as athlete's foot, yeast infections, and ringworm. Parasites cause parasitic diseases like malaria and toxoplasmosis, known as roundworm. Transmission of diseases can occur in different ways. The easiest way to transmit is direct contact. This can occur by contact with infected humans or animals, or from a mother to an unborn child through an umbilical cord. Indirect contact occurs when infection is spread to an object from an infected person and then later touched. Insect bites can also transmit diseases such as mosquitoes transmitting malaria, and contaminated food is also a common transmission of bacteria like E. coli and other diseases through contaminated food and water. There are 3 lines of defence to fight against foreign invaders. The first line of defence is non-specific, also known as the innate immune system. Its function is to defend against all pathogens in the same way and preventing invading pathogens from entering the body. These can include physical, chemical, and biological barriers, non-specific immune cells, inflammatory signal, or complement proteins. The skin is the body's largest organ and is an important immunological organ acting as our main physical barrier and primary defence against invading pathogens. It is part of the integumentary system and comprises of 3 main layers. The epidermis is the outer part of the skin that provides a waterproof layer and is made of epithelial cells and specialised cells called keratinocytes and Langerhans that prevent pathogens from penetrating the skin. The dermis is the middle part of the skin that makes up 90% of the skin's thickness, home to blood vessels and hair follicles. The subcutaneous layer, known as the hypodermis, is the deepest layer of the skin, mainly made from fat and connective tissues. Prevention of pathogens invading the body is usually effective due to the presence of mucous membranes. These membranes are coated in secretions. Whilst preventing invasion of pathogens, they also allow nutrients to pass through. They line the surfaces of the eyes, ears, mouth, nose, throat, sinuses, gastrointestinal tract, respiratory tract, urogenital tract, and vaginal tract. Other chemical and physical barriers include the gastrointestinal system, nasal cavity, airways, and the urinary tract. Females are prone to infection through the urinary tract. Although both males and females flush out bacteria upon emptying the bladder, pathogens are more likely to reach the bladder as females have a shorter urethra than males, making the passage of pathogens to the bladder more likely. Urinary tract infections are also more common during pregnancy due to an increased production of progesterone, creating relaxing of the urethral aperture, allowing easier access for pathogens to enter. The female genital tract, whilst healthy, has a pH of around 4.5. This prevents the growth of bacteria like bacterial vaginosis and maintains a protective barrier for the vagina. It is created by the secretion of lactic acid that is produced by lactobacillus. An inflammatory response is a second line of defence and also known as an innate response. It responds to harmful stimuli and commences a set of vascular and cellular events to initiate repair when an injury or infection occurs. Chemicals such as histamine and prostaglandins are released by damaged cells that send signals for vasodilation to occur, leading to increased blood circulation to the affected area. Gaps within the endothelial cells develop to allow large blood cells and fluid to pass through in a process called diapedesis. This causes inflammation. The area becomes swollen and warm to touch, with a redness in colour. This is due to the increase of blood and proteins attracted. Inflammation of the tissues can create pressure on nerve cells, causing the area to also become painful, and the inflammation usually continues for the duration of the infection until healing has taken place. A further second line of innate defence is a process called phagocytosis by types of cells known as phagocytes. This is initiated by the presence of macrophages at the infected area. Macrophages are formed through differentiation of monocytes and are a vital part of the immune system. They are large specialised cells that recognise, engulf and destroy pathogens and dead cells. Once a pathogen or dead cell has been engulfed, a vesicle is formed by the cell membrane. This is called a phagosome. A phagolysosome is formed when a phagosome fuses with a specialised cell called a lysosome. This contains hydrolytic enzymes to destroy the pathogen within the phagosome. This is the final stage within phagocytosis. There are more than 30 different types of complement proteins, including monokines, interleukin, interferons, and they are all types of cytokines. They travel in the blood, and once activated, they interact with each other to form a cascade of reactions. The reactions attract phagocytes to enhance the inflammatory response and phagocytosis. The complement protein C3b attaches to a pathogen, allowing it to be easily engulfed by macrophages, as it binds to a complement receptor 1, which is expressed on the surface of phagocytes. A pathogen is an organism that causes disease. Antigens are proteins that are found on the surface of a pathogen and are often required to infect the body. They communicate with the body that it is under attack to stimulate an immune response. There are three main types: exogenous, which is foreign to the host; endogenous, which is produced by intracellular bacteria and virus replicating inside a host cell; and autoantigens, which are produced by the host. Acquired immunity is not present at birth, it is acquired over your lifetime. When we are exposed to antigens, our immune system determines the best way to attack each antigen and develops a memory for specific antigens. This allows our immune system to learn how to detect, attack and destroy these antigens in the future. White blood cells, known as leukocytes, are usually referred to as one type of cell. However, this is incorrect, as there are 5 different types of leukocytes, all produced in bone marrow. These are neutrophils, eosinophils, monocytes, basophils and lymphocytes. Neutrophils are the most common type of leukocyte. They are a medium size with an irregular nuclei. They attach themselves to the walls of blood vessels to attempt to block pathogens from accessing the bloodstream. They are the first cells at the site of infection and destroy pathogens in a process known as phagocytosis. Immunoglobulins, known as antibodies, are produced by plasma cells and have many functions, such as targeting infection, recruitment of immune cells, and neutralisation of toxins. They are structured in a Y shape with typically 4 chains, of which 2 identical chains are lighter. They are categorised into 5 major classes due to the heavy chain that they have. The 5 classes are A, D, E, G and M. IgG is the most common. It is able to stimulate a direct attack against foreign pathogens and is the only type of immunoglobulin that can transfer from a mother to fetus across the placenta, providing some protection against unexposed pathogens. IgA plays a key role in cellular defence of mucosal membranes of the body, particularly in respiratory and digestive tract. The IgE are the largest molecule. They increase levels significantly in response to parasitic infections and allergic reactions. IgD acts as an antigen receptor on the surface of B cells, and IgM, they are the first antibodies produced in the immune response to invading pathogens. They play an important role to activate the complement pathway. Levels of IgM rise initially upon infection, but will begin to reduce when IgG levels increase. IgM comprises of 5 monomers within a pentamer structure. The body has the ability to distinguish between antigens that are either self or foreign invading. This determines if the body should therefore establish an attack. Nucleated cells are presented with molecules on their surface known as major histocompatibility complex molecules, class 1. These molecules allow the cell to be recognised as self and signals for the immune system of self-tolerance and will therefore not attack the cell. The immune system varies in its response to foreign antigens, as it does not have the same level of sensitivity to all antigens and will therefore initiate the strongest response if the antigens presented are different to those from human antigens. Hypersensitivity is an abnormal overreaction of the immune system to repeated exposure of the same antigen. There are 4 groups in which this is described. Type 1 is immediate. This is an immediate reaction induced by the secretion of IgE antibodies that can be seen in conditions such as bronchial asthma, food allergies and anaphylactic shock. Type 2 is antibody mediated. cytotoxic reaction mediated by IgG or IgM antibodies, for example, a reaction to medication or drugs. Type 3 is immune complex mediated, which is a reaction mediated by immune complexes, and this would cause conditions such as rheumatoid arthritis. And type 4 is cell mediated. This is known as delayed type hypersensitivity, and is a delayed reaction mediated by cellular response, resulting in conditions such as Crohn's disease and multiple sclerosis. Autoimmune disorders can affect almost all parts of the body. Common symptoms are fatigue, fever and inflammation, and affect areas, organs and tissues such as joints, muscles, skin and connective tissue, as well as endocrine glands, blood vessels and red blood cells. Rheumatoid arthritis is a chronic inflammatory condition that mainly affects the joints, causing painful swelling and joint stiffness. Early signs of the disease typically occur in the fingers and toes, but as it progresses can affect other joints like the wrists, knees and elbows. In severe cases it can cause bone erosion and joint deformity. Rheumatoid arthritis can vary in severity, and a person can have periods of remission and flare-ups. Medication and physiotherapy can ease symptoms. It is believed that women, especially middle-aged, are at a higher risk of developing this disease. There is also an increased risk of developing other conditions such as osteoporosis and carpal tunnel syndrome. Multiple sclerosis, known as MS, is another autoimmune condition described by the NHS as a condition that can affect the brain and spinal cord, causing a wide range of potential symptoms, including problems with vision, arm and leg movement, sensation or balance. It is the immune system attacking the myelin sheath that protects the nerves. The condition is most commonly diagnosed in people in their 20s and more commonly in women. Family members are also susceptible to inherit an autoimmune condition. There are two types of MS. The most common is relapsing remitting MS. Typically, a person will experience episodes of new or worsening symptoms known as a relapse. These can sometimes be triggered by illness or stress. Symptoms will improve over a period of time. Similarly to rheumatoid arthritis and all other autoimmune diseases, there are no cures. However, medication can speed up the recovery in between relapses, and treatment for individual symptoms can be managed. Transplantation is the transferring of cells, tissues and organs from a healthy person to another by using an organ donor. They are carried out to correct a malfunction of an organ system, for example, a heart, lung or kidney. Unfortunately, a transplant is not always successful. This is due to the ability within the immune system to recognise the antigens and molecules on the transplanted organ of the donor as not being self, and will initiate an attack as they're viewed as foreign. Reduced rejection of a transplant can be attempted by closely matching both the organ donor with the recipient with tissue typing. However, no two people except for identical twins can have identical tissue antigens. A medication containing monoclonal antibodies can be given during and after surgery. These are used to knock out specific T cells which can cause rejection. Three types of rejection are hyperacute, which occurs within minutes, usually seen if the wrong blood type is given; acute, which is usually within 3 months. All recipients will experience some amount of rejection and is therefore the most common; and chronic, which can occur over years as the new organ is slowly damaged by the immune system. Active immunity refers to the immune system producing antibodies as a result of exposure to disease. This can either be natural or artificial. Natural immunity is acquired from exposure to pathogens that cause specific disease through infection. The body will attack a pathogen it is exposed to and create antibodies. These antibodies will be memorised within B cells to provide long-term immunity to the pathogen in the event of future infection. Artificial active immunity is gained through a person being intentionally exposed to foreign antigens. This is presented within a vaccine to stimulate the immune system to create antibodies required to fight future infection in the same way as natural immunity. Both of these types of active immunity provide long-term protection. Passive immunity can also be gained via natural and artificial means. Natural passive immunity is provided when antibodies are transferred from one person to another, usually either via pregnancy when antibodies cross the placenta to fetal blood, or antibodies can be transferred during breastfeeding. Artificial passive immunity is immediate immunity provided by medication or a transfusion, where the antibodies are made by someone else. Protection in these ways only lasts a few weeks, as the antibodies have not been created by your own immune system. A vaccine is designed to prevent disease. They contain pathogens such as live pathogens, harmless fragments, toxins, and dead pathogens at a weakened or harmless version. But when the body's immune system is exposed to the vaccine, an attack will be initiated to produce antibodies and the production of memory cells to fight the pathogen. The two main types of vaccines are live vaccines, which is a vaccine using bacteria or virus that are weakened in a laboratory. A small dose is provided in a vaccination, which replicates in the body for the immune response to be stimulated. This response is almost identical to a natural infection. Inactivated vaccines are produced by growing the bacteria and usually require multiple doses. A protective response develops after the second or third dose, as the first only primes the immune system. Immunological memory allows the immune system to provide long-term protection against the infection throughout life. The first time an antigen is identified by the immune system, it is called a primary response. B cells identify the antigen for plasma to produce the appropriate antibodies. During this time, the infection can take hold of the body. Memory cells can recognise the pathogen, enabling them to produce antibodies quickly and for a longer period, fighting off future infections, usually before symptoms are even seen. This is called a secondary response. Vaccinations help protect against serious disease and are essential in the protection against viral diseases. They cannot be treated by antibiotics. Measles is described as an acute, highly contagious viral disease that can be contracted at any age, can be spread easily, and can lead to complication and even death. As you can see in the table, a steady decline in measles has occurred since 1940, and following the measles vaccination programme in 1968, a further considerable reduction was seen in the number of cases. The second chart shows confirmed cases between 1996 and 2017, with a substantial increase in 2012 with 2016 cases. These outbreaks of measles are believed to be due to a reduction in children receiving the MMR vaccine in the 2000s. Since the introduction of the measles vaccine in 1968, it is estimated 20 million cases and 4,500 deaths have been prevented within the UK. A BBC article published the 1st of February 2022 issued warnings that the uptake of the MMR vaccine is only at 85.5%, the lowest for a decade, and below the 95% target recommended to prevent future outbreaks. Coronavirus pandemic is a factor as to why the uptake of the vaccination has been lower. Although cases have plummeted due to social distancing, rising measles is expected when restrictions are fully lifted. There are many benefits of vaccination programs. Extending life expectancy can be seen in the COVID-19 vaccination program. Other benefits include economic growth due to poor health in unvaccinated people and cost saving within healthcare, as vaccination programs are much cheaper than the cost of treating disease. The concept of herd immunity is that if the majority of people are immunised, then the spread of the infection will be less likely, therefore preventing vulnerable from catching the disease. There are many reasons why herd immunity can be difficult to achieve. For example, some pathogens can have many different variations which prevent different antigens. This makes it difficult for the immune system and B cells to recognise all variants. The primary response will therefore take longer to produce antibodies, allowing an increase in transmission of the virus. It is also difficult to maintain herd immunity with a growing population, making it easier to spread infection with mass transport links that allow disease to spread globally, such as the current COVID-19 pandemic. Varied demographic also means that some people suffer more than others. Although many childhood diseases such as polio, measles, mumps and chickenpox have been eliminated due to vaccination programs, there is concern due to health scares and ill-informed information that parents received. For example, links between the MMR vaccine and the development of autism were raised in 1998 but later declared as fraud and blamed for lower vaccination uptakes, resulting in increased measles infections in 2012. Many vaccination programs exist for children and are carried out by health visitors, nurses or GPs during specific timeframes of a child's life, as well as other programs such as influenza and COVID-19 vaccinations that can now be administered in local pharmacies, allowing more flexibility in the availability of the vaccine and therefore increasing the uptake of such programs. Effectiveness of programs can be caused by the perception of some people that have an overall concern that vaccinations cause more harm than good, causing other diseases. They are known as anti-vaxxers.